Sorry, before we officially get started, did all of you see the assignment guideline for the sort of written assignment that I posted for you? No? Yes? No? It is up there under content, so you can go check it out. I'm going to talk about it really briefly. You guys read primary papers at all? Yes? What was the last good primary paper you read? What was it on? Or what journal was it from? Just out of curiosity. Yes? Okay, not bad. So is it Journal of Biological Chemistry?
I read a couple, like it was just all.
Okay. Okay, not bad. Have you guys heard of journals like Science and Nature or any of those? Yes? Good journals, bad journals? There are more handouts at the front if you need a hard copy of today's notes. So if you're just sitting up here, please help yourselves.
Remember the discussion on Monday about the so I spoke with the Facebook. They said it's pretty standard. I submit them and they tell you that I submitted them for them. And it's up to us to make our own recommendations. So we were okay. So you might need to consider waiting the first midterm, and so I don't have to write it because I'll be on vacation for 3-4 weeks. That sounds all right. Okay, so should I just email you or something like that? Or?
Well, they email me.
Okay.
You bring the note over to them.
Yeah.
They email me. Then we say, okay, this is what's going to happen. So I write you an official email so that you have a record of what's going to happen. So do I. That's it.
So I guess I'll ask you, like, what would be the combinations that we make, just so I'm aware? Like, how would you see that thing? So I'll submit it.
Yeah,
but
we'll just reweight it to the other. So you won't have to wait a makeup. We'll just have a little bit more on the next term test, a little bit more on the final exam, and then if you want, you could have a little bit more on your written assignment. It depends on you. Like, if you want to break it down for how you want to reweight it.
I think.
So if any of you are streaming Netflix on here, could you stop streaming during thing? I'll just ruin everything for you. Jon Snow is still alive. Don't have to watch it anymore. I just told you. But I think we're going to try to make sure that the internet bandwidth is okay, so that when we're streaming, that it's not cutting in and out.
Oh yeah, in the morning.
Okay, I think we're just gonna get started if you don't mind, and we'll see how things go today. I am going to try to stream this one more time, and we'll see how it goes, and we'll see how much bandwidth you're using up during the lecture. If it kind of slows down or it stops recording, then we'll know that there are issues with the internet in this room. So I'm going to actually pick up where I left off in the last lecture, and then I'm going to jump into sensory systems. And again, we really only have like 2 more lectures before the material that you're going to get on the term test is set. So after next Wednesday's lecture, we're going to have sort of a review session at the end of it, and then you're going to have the term test right after. And my job is to make sure that you're ready for that term test to the best of your ability and my ability to get you ready. Just before I go back over the last slide that I left off with on electrical synapses, and we go back to talking about the proteins that define electrical synapses, which are these connexons, just a reminder to go back and check over the assignment. I'm actually going to be a little flexible on the due date for the written assignment. It's worth a fair chunk of your mark. It's about 15%. I want you to do a good job on it. I don't know if you're in the habit of reading primary papers. I don't know if you understand the papers that you read, but that's my job to try to help you the best that I can. As long as the paper that you choose is not something that you've used in another course, the guidelines I've put out for you are for nature and for science. And that's really because the other section, they're using specific papers. However, if you're more comfortable looking at a journal like JBC, the Journal of Biological Chemistry, I don't even mind a journal like Journal of Nutritional Science, whatever it is, as long as it's related to a topic that you're interested in, and it's a topic that relates to human physiology and or anatomy, then I'll be okay with it. And one of the things that you can do is to bring your paper in, and I'll sit down with you and I'll tell you, this is a great paper, you should go ahead and start critiquing it according to the guidelines that are available for you online. So again, maybe that could be a piece of homework that you do over the weekend, just to find a primary paper published in the last 3 years that you feel you're interested in trying to do a critique or a review on. So I left you with these electrical synapses. We do not have very many electrical synapses within the CNS. The vast majority of the ways that in which neuron A and neuron B communicate with each other do not involve electrical synapses. These are really good for different forms of muscle, and we can identify these types of electrical synapses in cardiac muscle and smooth muscle, where the electrical activity, the ions, travel from one smooth muscle cell directly through these connexons into the neighboring smooth muscle cell. But we don't see very much of this in terms of the brain, which is what we've been looking at. And in the brain, one of the things that we see a lot of are what are known as chemical synapses. And these synapses are where we have neuron A connecting, or not connecting, but releasing chemicals onto neuron B. And so we have a presynaptic terminal from neuron A. There is a gap, which we call a synaptic cleft. And then there is neuron B, where the receptors for those neurotransmitters are going to be found. And one of the reasons why this process works the way that it does, and why we talked about the action potential in the very last lecture, is that action potential is actually really important, that once it reaches the end of the axon, at the end of the axon, we have a specialized compartment where we have these synaptic vesicles. These are little packets that contain neurotransmitters, and when that action potential reaches the very end of the synapse at the presynaptic terminal, it causes voltage-dependent calcium channels to open. Calcium comes in, and calcium will then cause the neurotransmitter vesicles, neurotransmitter-containing vesicles, to fuse or bind to the presynaptic terminal and release their contents into the synaptic cleft. They will then travel across and activate the postsynaptic membrane receptors. And I'm going to give an example of what happens in a chemical synapse. So we spent most of the last hour in last lecture talking about action potentials, how we have these voltage-dependent sodium channels that cause the electrical activity that we started off with at the soma, how it travels down the axon. And we talked about 2 different ways that electrical activity travels down the axon, these action potentials. Do you remember the 2 main ways in which it just travels down? A lot. Do you remember that one? Do you remember the word that we use for jumping? Yes. So we talked about saltatory conduction, thank you very much. And the saltatory conduction at the nodes of Ranvier, between where myelin is wrapping, is one way that we spent talking about how action potentials will eventually reach the end or these terminals. Once the action potential comes down to the ends of these terminals, one of the things that it can do is to cause these voltage-dependent presynaptic, because this is the presynaptic neuron here. Here is the synaptic cleft, and here is the postsynaptic membrane down here. What happens is the calcium then comes in after the voltage change opens up these channels. Calcium then binds to these vesicles, and it causes these vesicles to then move and bind to the membrane, the presynaptic membrane, and it spills out its contents into the synaptic cleft. So these chemicals are released here after an action potential, and then theoretically they can then bind, these neurotransmitter molecules can then bind to the appropriate receptor sites. So think back to the last lecture. I told you we have voltage channels, we have channels that are just like pores, which are open all the time. And then I told you we have these ligand-gated channels. Here, these ligands are those neurotransmitters, and when it binds, it opens up the channel, and in this case, it just happens to be, so this will then cause the sodium to go into the cell, causing depolarization. Is that okay for everyone? Is there any step in this process that you'd like me to go back over, or you're not 100% sure of? No? In order for this to be a real signal, though, in order for that neurotransmitter to actually code for a real signal that something is going on, that neurotransmitter can't stay there forever. So imagine it stayed there. The signal that caused this action potential to go down is long gone, but that neurotransmitter hangs around. If it did that, you wouldn't actually be able to encode any information. It's like doing this. So I touch the wall, I move my hand away, but if these neurotransmitters were still there, it would feel like I'm still touching the wall, even though my hand was moving away. That signal has gone away, and so has the neurotransmitter has to do this as well. And there are two different ways in which we can remove these neurotransmitters, and it's summarized on that previous diagram, which is, or that previous slide, and I'm just going to go into this in more detail. And I talk to you about two different neurotransmitters. One is acetylcholine, and then the next one is noradrenaline. And acetylcholine is one way that we can stop this neurotransmitter from sticking around this synaptic region, to stop it from binding to and activating these particular channels. We don't want it to be there forever. We want to have it there when a signal comes down, it does its job, it transmits to the next neuron, but if it stays there, it's not very beneficial to us. So once it binds to receptors and then it starts unbinding, in theory, it could go back and bind again. But there are mechanisms that are in place, including enzymes like acetylcholinesterase, where once it's released, it will actually start to break these molecules down. So it means that over time, once the neurotransmitter has bound, done its thing, opened up sodium channels, and then has been released, acetylcholinesterase will break it down, so there's no more opportunity for it to go back and bind again. In other words, we have that removal mechanism that we needed to have. So it breaks acetylcholine down into its components, acetic acid, and then choline. Your brain actually is really efficient. It likes to reuse things as much as possible, and so the choline is actually taken back up into the neuron, and it is repackaged and resynthesized back as acetylcholine into the synaptic vesicles. Are you all good with that? Yes? Okay, so yeah, do you have a question?
Yeah, so we break down ACH outside in the synaptic cleft, and then that is sent back to the
So the acetylcholine is broken down into its component parts, acetic acid and then choline. So both of them together make up acetylcholine, and now we've just broken it down. So there's no more ligand around.
So what happens to the acetic acid?
The acetic acid is actually taken up not by neurons, but by the surrounding glial cells. and the glial cells will then use acetic acid as another form of energy. You don't need to know that for your term test or anything else, but I thought I'd answer your colleague's question. Another mechanism to remove these neurotransmitters from the synaptic cleft is a very common mechanism, somewhat similar to acetylcholine, but it involves a different molecule. So when you see acetylcholinesterase, it makes sense that you are dealing with the breakdown of acetylcholine, right? But here it's a little bit different. So just like we had before, we have a neuron, a presynaptic neuron, packets of neurotransmitters in the synaptic vesicles. Calcium causes it to bind. It releases norepinephrine into the synaptic cleft. It binds to norepinephrine or beta adrenergic receptors over here on the postsynaptic membrane, and it will cause this neuron to change its potential. However, unlike what we saw with the acetylcholinesterase, we don't have that sort of norepinephrase sitting in the synaptic cleft. We still have to get rid of norepinephrine. The mechanism here is a little different. The mechanism that we use for norepinephrine is simply to take it back up into the presynaptic terminal. So it gets taken back up, and then it can either be repackaged back into a synaptic vesicle directly, so it's not broken down, it's just simply taken back up, or there's another mechanism here that is being shown called MAO, and we have different types of enzymes, MAOA, MAOB, and these monoamine oxidases, which are enzymes that are found in neurons, could also inside this neuron break down norepinephrine into inactive metabolites. Okay, so we have two different, very different. One is to in the synapse, and this mechanism down in the presynaptic terminal, not in the synaptic cleft. Is that clear for everyone? So again, the hard thing here is to keep clear that monoamine oxidase, unless you're taking pharmacology courses, and it's something that you'll hear lots about, monoamine oxidase is the enzyme that will break down things like norepinephrine. Do you know any other neurotransmitters that MAO breaks down? Maybe you've heard it in other psychology courses, in any other physiology or biochemistry courses, anatomy courses. Yes? You can guess. There's no harm in guessing. Sorry, GABA? It's, don't guess like that on test. Okay, yes? Serotonin? Yeah, there is some evidence to suggest that serotonin can be broken down by monoamine. What's another neurotransmitter that contains the word amine? Dopamine. So dopamine is another way to, or another neurotransmitter that's broken down by monoamine. So this is a way in which neurotransmitters don't continue being left out here in the synaptic cleft. Do you see an issue with that at all? Like any problems that you could see physiologically, like with this, or how you might use this to your own advantage? And some of you might be interested in doing this for your literature review or your critique. So what's one theory about what goes wrong in your brain when you are depressed? So one hypothesis is, and it's a neurotransmitter hypothesis, is that somehow in depressed individuals with depressed neurons, there just isn't enough of this neurotransmitter to go around. One of the old class of drugs that was an antidepressant is a monoamine oxidase inhibitor. So it blocks the breakdown inside the presynaptic terminal. So if in depressed individuals, neurotransmitters are down, and this is a way to break down the low levels of neurotransmitter, antidepressants make sure that we, in theory, have enough of the neurotransmitters around. Again, it's just a theory. We don't actually think neurotransmitter levels in depression are all that important, to be quite honest. And it's something, again, that you might want to go and do a literature review on. So this gets into this next sort of phase, which is we're talking about these different receptor molecules that are found on the postsynaptic membrane, acetylcholine receptor, acetylcholine receptors, or we could be talking about serotonin receptors, or we could be talking about dopamine receptors, etc. Your textbook actually says that neurotransmitter only fits in one receptor. Is that true? Can you give me an example where this is not true? And it turns out that it's not true. For example, if we go back here, norepinephrine can bind to more than one type of receptors, not just one receptor that norepinephrine can bind to. Anyone have any ideas about other receptors that you've heard about? Because we're going to talk about it in a few moments. So if you were to tell me, and this gets back into finding out how much you already know in terms of your biology, what is the most common neurotransmitter in your brain? Every last one of you in this class has this neurotransmitter, like it's the dominant one. What do you use as a neurotransmitter? The most common neurotransmitter in anyone's brain, what is it? Not serotonin. We really have to talk before you write your term test. Okay, always go with B or C. That's one. And then if you're going to guess, don't go for serotonin. Yes. Acetylcholine, not in the brain. No. Any other guesses? Yes. Dopamine, not dopamine. Yes. Adenosine, that's a good guess. It'd be really good for glial cells, but not for neurons. Anything else? So have you heard of glutamate? Ah, yes. So 80% of your neurons actually use glutamate as a neurotransmitter. We're going to talk about glutamate. But in fact, glutamate as a neurotransmitter can actually activate many, many receptors, not just one. There's no one glutamate receptor. Okay, not all cells have receptors, very true. So not all neurons have receptors, not all glial cells have receptors. The neurotransmitters that are released, glutamate, for example, is excitatory in most cells, but in other cells, it can actually turn off the activity in those cells. It can actually look like it is inhibitory. I know it's a very hard concept to think about right now, but we'll come back to that a little bit later on. Okay, so we're going to talk a little bit more about neuromodulators very, very briefly in this context of we want to understand how synapses work. In your brain, for you to be able to do everything that you do with your brain, firing action potentials by itself is useful and it's necessary. But in fact, in order for your brain to adapt, we need to think about how the different neurotransmitters and everything else can change the overall activity in your brain. And this gets into this idea of neuromodulators. And before I get into neuromodulators, where sometimes a neurotransmitter actually enhances the amount of neurotransmitter being released. Sometimes it decreases the amount of neurotransmitter being released, which is what this whole slide is saying. Before we get there, we have to go back and read these postsynaptic potentials. And when we talk about postsynaptic potentials, we're talking about measuring electrical activity in this cell. And we're talking about this resting membrane potential. When that neurotransmitter that we just looked at has crossed that synaptic cleft, glutamate or some other type of neurotransmitter has bound. If we were to measure the change in the resting membrane potential, we might see this depolarization, which means it's getting closer to this threshold value or becoming more and more positive. If it reaches threshold, this neuron that is receiving neurotransmitters might fire an action potential. That means neuron A has been successful. It has conveyed information to neuron B, which is where we're measuring now, and it will then fire an action potential in return. In some cases, we might actually want to turn things down. We might want to turn off activity in the brain, and sometimes the neurotransmitter that gets released will actually cause an inhibitory postsynaptic potential where it's hyperpolarizing or moving farther away. It's this neuron is now less likely to fire an action potential. Okay, and this hyperpolarization is generally an inhibitory response. Is that okay for everyone? Normally, the neurotransmitter that we talk about up here is glutamate. Glutamate is usually, in most cases, like the vast majority, 95% of the time, it is going to cause an EPSP, or an excitatory postsynaptic potential. Can you tell me a neurotransmitter that you might have heard about that might cause an inhibitory postsynaptic potential? So an action potential has come down neuron A. It has caused calcium to come in. Little packets of neurotransmitter have been released, and when pressure from neuron B, actually see neuron B becoming hyperpolarized. What was the neurotransmitter that got released that caused this type of activity? Do any of you know? Shall we make this a little more interesting? Yes. GABA, yes. So GABA is usually an inhibitory neurotransmitter. So if 80% of your brain is glutamatergic, excitatory, 20% is usually considered to be GABAergic, or inhibitory. You only need a little bit of inhibition, in other words, to control all the functions in your brain. Now, I'm not going to go over this in a lot of detail. I actually have highlighted what I'd like you to make sure that you know for your term test. Some of you who have taken neuro courses, psych courses, will have heard of this a lot, and some of you will be hearing about this for the very first time. And I'm going to tell you what you need to know from this particular slide. And this is a slide that I'd like you to make sure that you know. And I have to apologize, I don't have a little picture of Drake, and when you see the lecture notes for next week, you'll actually see someone else sort of taking center stage next week. And when you go back over your notes, this is a slide that I'd like you to spend time reviewing over the weekend. So I don't need you to know the different anatomical terms for axonic synapses. Axonic just means that this is an axon, this is an axon, and we have two axons that are making connections with each other. But I don't need you to really worry too much about this. These axoaxonic type of synapses, though, these are both making or are going to modulate, change the activity of this presynaptic release. So under normal conditions, for example, you have an action potential coming down that we've been spending the last few minutes talking about, and when the action potential comes down, it activates voltage-dependent calcium channels here. Calcium then rushes into the cell. These vesicles then bind to the presynaptic membrane, causing the release of all this neurotransmitter. This neurotransmitter then binds to receptors that we find here on the postsynaptic membrane. But we can change that. So this is the normal situation here on the left, okay? But we can change this. Let's imagine for a second that one of these axonic synapses, so again, forget the details of this, a synaptic contact, presynaptic terminal. One of the things that might happen is, let's say this is an inhibitory axon, okay? It causes inhibition. And one of the things that can happen is that, let's say, when this neuron is active, so it's firing an action potential, at the same time another neuron somewhere else in the brain is trying to turn things off. Like you've just made a mistake. You were going to go and pick this up, you decide not to do that, you want to stop that activity from occurring. So imagine this inhibitory neuron, this is fired and this is firing at the same time. It's inhibitory. So one of the things that happens is that this will actually change the membrane potential here so it doesn't depolarize as much. The action potentials aren't as effective, and you can see that there is less neurotransmitter being released. And this one diagram that you see here in panel A, which is a normal situation, and when we have this inhibitory neuron that is causing an inhibitory postsynaptic potential here, this neurotransmitter is being released, and we call this presynaptic inhibition. So presynaptic now, this inhibitory firing at the same time as this, the total amount of neurotransmitter that is being released from this presynaptic terminal, instead of being this huge amount that we would normally see, is this reduced amount. Now, why is that important? So, for example, we think this is a way in which endorphins are actually able to inhibit pain sensation. Where do you normally associate endorphins? Where have you heard about endorphins before? Or have you heard about endorphins before? Yes? It's very much like a drug, yes?
Like, is it heroin that modulates what endorphins do? And that's why you feel the way you do?
Maybe. So you're kind of on the right track. Yep. So if you're a long distance runner, you can tell I'm like really right. Why are you laughing? I'm actually sucking my gut in right now. Um, if you're a runner and you're going for a really long run, let's say you're going for a marathon run. I've never done it, but I've heard it's like really painful. But after a while, individuals that are running, their muscles really hurt, etc., etc., the endorphins kick in. They get this runner's high. And these endorphins actually inhibit that pain response that is being felt up in the brain. And so we know these forms of presynaptic inhibition are really important. And endorphins are sort of the naturally occurring opiates or opioids that we have within our body. Opioids, the same types of opioids that you would take if you have pain. And they block pain just like morphine or heroin or other types of things do. We can block presynaptic release, but we can also make sure that we get even more neurotransmitters being released. We can facilitate presynaptic neurotransmitter release from these presynaptic terminals. And one of the ways in which we can do this, and I'll come back to this later, but for the time being, I'd like you to make sure that you understand that glutamate being released from this type of synapse as an excitatory, no longer inhibitory, excitatory, might actually cause more nitric oxide to be produced by this particular neuron. Now I'm going to come back to that. We haven't talked about nitric oxide. Maybe you've heard of nitric oxide sort of like as a gas in other lectures or in your chemistry course, but but neurons themselves are capable of producing gases that work very similar to neurotransmitters. Any questions about this before I go on? No? Okay. So I'm not going to ask you to know all of the different details that are on this particular slide. What this is suggesting, and the next few slides are going to go over the same sort of concept. We're looking at this one cell. This one neuron is actually receiving multiple inputs. So you have more than one presynaptic terminal coming down. It's not that simple case that we looked at before. And it's different than what we just looked at with presynaptic inhibition, and it's not the same as presynaptic facilitation. That's all at this level at the presynaptic. What we're looking at is what happens when we have like one neuron coming in and another neuron coming in, they're releasing neurotransmitter. What happens overall to this particular neuron? In one scenario, if these are both excitatory and they're both causing depolarization, if they're close enough together, remember postsynaptic potentials can summate, they can add on top of each other. If they're spatially close enough together, then it might mean that when they sum or when they add to each other, that it might be big enough to reach threshold at this trigger zone to cause an action potential to fire. So spatial summation is one way to activate a neuron, in other words. Another way is that's if you have more than one input. And again, I'm going to ask you a question related to either spatial, temporal, etc. Spatial, you always have to have more than one input. So if you see if you see like multiple presynaptic inputs, you know right away it's spatial summation. If you only have one axon or one presynaptic neuron coming in, then we have this different scenario. So there are no other synaptic inputs coming in, it's just one. And what this is suggesting is, in order for this neuron here to start firing an action potential, what has to happen is this presynaptic neuron has to fire really quickly in a very short period of time. And this is known as temporal summation. These two action potentials fire very close together. And by firing very close together, it causes enough of a great potential to cause an action potential to be generated at this trigger zone. So again, to make it clear, temporal summation involves one presynaptic neuron firing very close together. Spatial summation, two or more neurons are close together, both excitatory, being or activated at the same time. So on your term test, would you be able to distinguish or be able to remember those two different types of summation? Yes? No? I guess we'll find out in about 2 weeks. Okay. Again, other scenarios, and I'm not going to try to trick you up or trip you up or do anything like this, but you can imagine that this is the simplest case in the previous two diagrams. This is actually the real life scenario. This one neuron is actually going to have multiple inputs. Some of those inputs are going to be firing close together, that same input. This would normally be temporal summation. Some are going to be inhibitory, some are going to be excitatory. And what this neuron has to do is figure out, based on all of these different inputs coming in, is it enough for me to reach threshold and then fire an action potential if it reaches threshold at the level of the trigger zone? Any questions on this before we switch gears a little bit? No? So I'm going to ask you on the weekend, and I'll make sure that we review this before you go today. And again, I'm not trying to cover all the slides. I'll try to get through as many as I can, but if we leave you, don't panic. I'll make sure that whatever you know for your term will be there. I was asked a question earlier, are we writing the same term test as the other section? It'll be similar, but it'll be based on the material that we cover here. We're going to be sharing about 2 or 3 questions in common between the courses, but overall I'm going to be writing a separate set of questions for you, and I think Dr. Agard will be writing a separate set for her own class as well. So I want you to find a paper that you're comfortable with. Doesn't have to be Nature or Science. Go through the sort of assignment guidelines. I posted a rubric for you as well that breaks down like how you're going to be evaluated. And then over the weekend also, I'm going to be posting some sample multiple choice test questions that will be at exactly the same level of difficulty that you'll get on your term test. I want you to try them. And if you don't understand how the answers are derived, or you're not sure like where you've gone wrong, then I want you to talk to me on Monday. Okay, so those will be available for you sometime over the weekend. You'll have all weekend to look at them right up to the beginning of class. So I want you to start going over things, review early and often. We will go through a lot of material. In fact, we've already gone through over, I think now about 100 and some odd slides. That's a lot of material already. I'm going to go through almost another 100 today if we're on track. So it's a lot of material. Review early, review often. I'll try to point out things specifically again in today's lecture that you should go over as well. Any questions or comments, suggestions before we go on? Now everyone looks really scared. You're not scared, right? Term test, be a breeze, easy peasy. Yeah? Okay, good confidence. So I want you to think about sensation. So our brains process everything that our body encounters on the surface. So if something is touching your skin, you're aware that something is touching your skin. Without even without opening your eyes, you'll know that your leg is like bent in space. It's able to have all of these different types of sensations. Your brain is able to do a number of different types of coding for different types of information. All of that involves action potentials, and all of that involves very, very specific signals being sent along very, very specific nerve tracks. So the visual information, for example, is kept separate and distinct in most people's brains than the nervous, the impulses coming in from the auditory system. Your hearing and vision have separate wiring systems, in other words, in the brain. Is that okay for everyone? But I also want you to think about it this way. What we're going to be talking about in the next few slides as we go through the different types of sensation, you guys are all capable of detecting light. So you can see light up here, right? Yes, no? Otherwise, I've been lecturing with pointing at things completely at random. But think a little bit about this. You're able, your eye, the optic nerve, the optic tract, which is specialized for vision and light, has somehow taken light energy and converted it into something your brain understands. So think of it another way. If I took your brain out and I put it there on the table and I shone light on top of your brain would not know that there's light shining on it. There is a system to transduce, to change a different form of energy into electrical activity that your brain recognizes as light. I could, in theory, if I was really, really mean, stick an electrode at the back of your head and stimulate the occipital lobe directly and cause you to see things even though it might be a completely darkened room. Our brain works to change energy, whether it's light, sound energy, etc., into something the brain can recognize in terms of sensation. All of it is electrical energy. It's all encoded at the level of action potentials, and those action potentials have to be decoded by the cerebral cortex or the brain. So again, I don't want you to bother trying to memorize all of this. We're going to go through an awful lot. I'm not going to try to trick you on little details that are found in these background slides, and the next few slides are all background slides. I'll tell you exactly when you should be paying more attention. So we have different types of primary receptors that can convert those types of sensations into things that our brain can understand. Like if you poke the brain, and I've been really lucky enough to be in the OR during neurosurgery, and you can poke the brain and the patient's awake and you can ask them, do you feel anything? And you're poking the person's brain, not that hard, but you know, you're poking the brain. They don't know. The brain can't feel anything. But we have this ability to take primary receptors, information on the body, on the surface of your skin, and convert it into something that lets the body know about there's pressure on the skin. You're touching something, that's pressure. You know how cold something is or how hot something is. You can also tell about joints and how they're located in space. Like, is your arm, without you even looking, is your arm up? Is it down at your side? Is it sticking straight out? You're able to tell about how your joints are moving in space. And of course, there's always information about pain as well. You also get information, and it's not always as obvious, but you also get information from your internal organs as well. It's a little bit harder to describe, and we'll come back to this in the second hour, that also tell you about whether or not you have pain in different internal organs as well as pressure. And then we have this other category of special senses. So your body has to take a chemical and change that into electrical activity when you are tasting something. When you smell something, same idea. You've detected a chemical out there in the environment. It's somehow bound to a receptor that has been converted to electrical activity. Your brain has to decode it as, oh, I remember that smell. That's the smell of the meatball marinara from Subway subs or whatever it is. So you have to be able to take these primary receptors and convert them into a form of electrical activity that your brain can recognize. So we're going to talk about these special senses, and again, don't bother trying to memorize this. It'll hopefully make sense to you. These special senses, and I'm going to try to get as far as I can. I may not make it to the visual system, and I'm going to try to get back or maybe stop it around the auditory system. And then there are other general senses about your sense of touch, things internally in terms of the visceral organs and how they feel pain as well as pressure. And sometimes that can be a real issue, and hopefully that one slide that I have in here will give you some ideas about this. Don't bother memorizing this, but it is a really good resource. As I said, I don't want you to buy the textbook unless you already have it. This is a really good sort of review if you need to go back like really quickly to go and check out what types of receptors did we talk about. This is a good summary. You don't have to memorize it. You don't have to worry about it. If you don't have the textbook, I put it on there for you so that you would have this sort of as a form of reference. So the different types of sensory receptors that we have are classified according to the different ways in which we sense things. And this is a good sort of summary slide for you to maybe circle and come back to over the weekend. So we have a number of different mechanoreceptors. We have all sorts of mechanoreceptors built right into our skin. So these are really good at detecting whether or not we're feeling pressure. Pressure equals mechanoreceptors. When we are compressing mechanoreceptors, we can detect that. If we're bending mechanoreceptors, and you'll have to think a little bit about this, but in your ear, for example, you're actually bending different parts, different mechanoreceptors that allow you to hear. You might be stretching cells in some instances, and again, these mechanoreceptors will allow you to tell if you are changing their activity that way. And all these things are going to tell us, these mechanoreceptors tell us about touch, about whether or not you have pressure on your skin, whether you have joints moving, whether it's to hearing, or whether it's related to balance. And again, for ND type mechanoreceptors, it's really hearing as well as balance. When we smell things or taste things, chemoreceptors, chemicals bind to receptors, and then they get converted into electrical activity. Thermoreceptors that we have in our skin, you have them in your mouth, etc. These also respond to changes in temperature. And then photoreceptors that we have in our eye respond to light and is involved in our visual system. Nociceptors are kind of a weird classification, and I'm going to go through that in some more detail. And these detect when you have really extreme pressure, really extreme mechanical deformation, whether we have chemicals being applied to the different receptors. If the range of temperatures is too high, you'll feel pain. And it's still a thermal receptor, but it's outside of its normal range, then it becomes a nociceptor. So again, let me ask you a question. How hot does something have to be for you to actually feel like it's painful? So you have a hot cup of coffee. Some of you are really, really good at holding a hot cup of coffee. Some of you need to ask the person at Tim Hortons or Starbucks, like, can you put it in another cup? What temperature is painful for most of us? When do you start feeling pain? Is it at 100 degrees where you're literally like your skin is like boiling? It isn't. But what temperature do we actually start to feel pain? When is heat, not like boiling heat, but when is heat actually painful for most individuals? Any ideas? 50 to 60, exactly right. Again, we have this idea that, you know, it's like 100 degrees. It's got to be like boiling hot in order for us to feel pain. But in fact, most of you will feel uncomfortable enough holding a cup that's at 60 degrees, not anywhere close to 100, where it feels painful to your touch. You've activated nociceptors, which are thermal receptors that are outside the normal range that thermal receptors like to be activated at. So again, keep all of this in mind. This is just for us to do some housekeeping. I'm not going to be asking you any questions related to this slide. It's just a different way to talk about receptors instead of how they respond to different sensation, which is what we talked about here. We identifying them or classifying them according to how they get sensed. But if we talk about where they're located, we talk about exteroreceptors on the skin, visceroreceptors that are found in the internal organs, and then proprioceptors, which we've sort of talked very briefly about with joints and tendons. But it's for your own information. Don't bother memorizing this, because I'm not going to go back and ask you a specific question related to this. Now, this is a good summary diagram, though. Now, I am going to ask you to make sure that you spend some time going over the next few slides. This slide encapsulates everything. This is a patch of skin, and this is the skin, the epidermis at the top in dark pink. And then in purple, we have the dermal layer. And you can see that most of the receptors that are found in your skin are not found on the epidermal layer. They're sort of found either at the junction of the epidermis in pink at the top, or found at the level of the dermis. Okay, we don't have a lot of sensory endings directly on the epidermis, your skin, but in the dermal layers underneath, we have a lot of them. So I'm going to come and talk about each of these in turn, but I'm going to highlight things right now so that when we go over the next few things, that you have a good sense of what I want you to make sure that you know. In your skin, we have two types of receptors. We have ones where the nerves are just like naked. They're called naked free nerve endings. There's nothing else associated with them. It's literally like having the nerve terminal just sitting there in your skin. So imagine like an axon coming up, which you see here in yellow, and then the terminals are up there directly touching your skin. Nothing else associated with them. That's a free nerve terminal. We use free nerve terminals to detect temperature and pain. Okay, so when we see pain and temperature, it's always a free nerve ending, always. When we talk about pressure on your skin, then we are looking at these other types of specialized cells that go along with it. So here is a Pacinian corpuscle, and the nerve ending is actually found embedded in this lamella, which is like an onion-like structure, where we have other keratinocytes that it has to compress first in order to activate the nerve cell. It's not a free nerve ending. Neither is a Ruffini ending. No free ending here. It's encapsulated in this Ruffini end organ. The Meissner corpuscles, a corpuscle is the body. So there are no free nerve endings. They're encapsulated in another corpuscle. So we have free nerve endings, which are responsible for pain and temperature, and then we have these other encapsulated nerve endings that tell us about pressure and different types of pressure on your skin. And then we have these last type that we'll talk very briefly about, the hair follicle receptor, but for a variety of reasons, I really don't like talking about hair follicles and hair follicle receptors, so we'll just bypass that one. Okay, so these free nerve endings, there's a lot on this slide, and this is a slide I'd like you to make sure that you know. So there'll be like 1 or 2 multiple choice test questions related to what's coming up over the next few series of slides. These are the most common type of sensory receptor that we have in the skin. We have a lot of them in our skin. Your skin has to be able to detect if there's something painful, that it's able to tell what temperature something is, and so we have a lot of these. They're scattered throughout most of the body, and again, this is that other type of where it's located. It's also found in the viscera or the internal organs as well. Again, another fact that I'd like you to make sure that you know, we have a number of things. We have different nerve endings that can detect cold, and we have another set of free nerve endings in your skin, and they could be side by side, but we have another set of free nerve endings that involve thermoreceptors that are there to tell us a little bit about warmth. But the interesting fact is the vast majority of your skin is actually embedded with free nerve endings that are there to tell you if you're getting too cold. It doesn't really tell you if you're getting too hot. They certainly will tell you about that. We do have warm receptors, but it turns out that our skin is actually adapted more to tell us when we're getting cold. We have 10 to 15 times more of these cold, free thermal receptors in our skin than we do the ones that detect warmth. Okay, and again, the pain-free nerve endings are there when we go outside these ranges. When we go beyond sort of the 50 to 60 degrees, when we start entering like 70 degrees before we can cause physical damage to our skin, where things start boiling, this is actually really important. You can also freeze your skin, so this is where these thermal receptors or free nerve endings become important. So these Merkel discs, again, just to keep things in mind, one of the places that we find this is right at the base layer of the epidermis. So it's right at the bottom of the epidermis, right under where your skin stops or the epidermal layer stops. And again, it's really good for detecting where you have light touch or superficial pressure. So if something is like lightly brushing up against your skin, your Merkel receptors are the ones that are getting activated. My wife is not a neurobiologist, nor is she a physiologist, and so she doesn't really get it when saying, would you like me to go and activate your Merkel discs for you? Want to go and caress her cheek, for example? One day she'll get it, hopefully. But this is that light touch, superficial pressure where you're just like lightly touching someone's skin. Again, we won't talk very much about these hair follicles, but when you look at your hair follicles, it's a little easier to look at my hair follicles, perhaps a little more spaced out. But when you look at the bending of the hair, because of the way that the nerve ending is wrapped around it, it actually detects when you have the hair follicle itself moving. And so it becomes very, very sensitive where you have skin that has hair embedded on it. You'll have a lot of these hair follicle receptors. So this is one that I want you to make sure that you spend some time going over. The Pacinian corpuscles are the deepest ones that we have in our skin. So in your hands, for example, we typically find them embedded in places like the palm of your hand. And these lamellated corpuscles and lamella, different layers, in other words, layered corpuscles. What is found embedded in it is one single dendrite. Okay, and then we have this onion-like structure underneath. So when you're pushing down on your skin, you actually have to push also on these layers in order to activate that dendrite. Remember, mechanoreceptor responding to mechanical changes. We have to be able to go and change that. So what type of stimulus, unlike the Merkel discs or tactile discs where it's like light touch, what types of things might activate these Pacinian corpuscles that are so deep in your skin, not at the surface, but much deeper, and also protected by these really layered onion-like structures. So this is where you have like really deep cutaneous pressure, like when you're really pushing on something, or when you have vibration going on. Okay, so you need to think a little bit about this because it's a little counter-intuitive in some ways. Can you tell me like when you might actually have pressure on your hands? So for sure, if you're leaning up against something, you are activating your Pacinian corpuscles, definitely. But can you think of other times where you have vibration on your hands? Where do you have vibration on your hands? Actually, I don't want to know. But can you think of where in a normal physiological condition you have vibration and no construction stuff, nothing else? So no jackhammers, nothing. Just when do you actually have vibration on your hands? Yes.
Lifting more than you can carry.
Lifting more than you can carry. Maybe. How do you think that that causes vibration?
You can take it so your hands start shaking.
Oh, your hands start shaking. Okay. Yeah, maybe. I hadn't thought of that. Yes? Sorry? Your cell phone. Okay. Yeah, maybe. You could have your cell phone set to vibrate. Definitely would happen. Yes.
When there's a lot of music playing and there's vibration.
Okay. So vibration from music, definitely the case. I'm going to tell you where normally you have vibration, where you actually have this under physiological conditions. It's not necessarily like that deep cutaneous pressure where I'm standing up against something and that's activating my Pacinian corpuscles. If you look at your hands actually have ridges on them, like you have your fingerprints. And when you move your hands across that your other hand, actually that's a form of vibration. You are stimulating these Pacinian corpuscles in that way. And these ridges, when things are slipping out of your hands, are ways that we can also activate these Pacinian corpuscles as well. And you might not think that it's so deep in your dermal layer that you might not be stimulating them, but they're sensitive to vibration, not just a mechanical force itself. I think now is actually a really good time to take a break. I'll continue on with the Meissner's corpuscles, which you'll also, I'll ask you to make sure that you know. So for that previous section, you should make sure you know the Merkels, the Pacinian corpuscles, and I'll ask you to make sure you know Meissner's as well. And then we'll come back and we'll go on with the rest of sensation.
Oh, I will be sorry. Visceral receptors. you mentioned how,
You actually look at the whole lecture on your phone?
yeah.
Wow.
Oh, is that?
No, I've never seen it. So that's
Oh,
cool.
I just got lazy being on my computer, so I'm like, whatever.
Yeah.
Yeah.
As phones get bigger, I guess it's possible.
Yeah. So, visceral receptors. It's just like associated with organs, but then in the next line it says scattered throughout most of the body.
Yep. Including things like
Did you recently get sick, by the way?
sorry?
Did you recently get sick, by the way? I noticed you're coughing.
I was, but not. I think I'm better.
Oh, so you're sick? A bit?
It was a long, like 2 weeks ago.
Okay. All right. I'm just curious, that's all.
Okay.
It doesn't really.
Okay.
Thank you.
Yep. No problem. Yeah. Okay, folks, if you don't mind, I'm gonna try to get going. Hey, folks, if you don't mind, I'm gonna try to get going. And we still have a number of different sensory endings in the skin to get through, including these Meissner corpuscles as well. And these are really important when you need to be able to discriminate two different points that are found on your skin. So this is what is being shown here. You can take a pair of calipers with pointy ends. You can set the distance apart from them. And if you have a lot of individual Meissner corpuscles, then you will be able to detect at the same time that you have two different probes touching your tongue. Even if they're like only 2.2 millimeters apart, you'll still be able to detect, I have two different things touching my tongue if you have a lot of these Meissner corpuscles in terms of this 2-point discrimination on your skin. And again, your tongue is considered part of your skin as well. On your fingertips, we also have a rich density of these Meissner corpuscles as well. And again, you keep your eyes closed. The investigator moves the caliper points to different distances, and they'll move them closer and closer together. And at one point, for example, somewhere between 2 and 3 millimeters, when they ask, like, how many different points can you feel, you'll say 1, even if it is actually 2 separate points separated by 2 millimeters. How do we know that this actually works? On your back, where you don't have many of these Meissner corpuscles, when you take these 2 different points, and it seems like it's bizarre, but if you were to take these 2 different points and then you were to ask, how many points do you feel on your back, the individual would say 1. Okay, because we don't have enough of these Meissner corpuscles, it can't detect it. It's just activating like this one group of Meissner corpuscles here through mechanical deformation. And so the person says, I can only feel 1 point on my back. And that's the difference between different parts of your skin. Some parts of your skin have lots of these different receptors, and some parts of your skin do not. And this is highlighted in this one example. So Meissner corpuscles, again, make sure that you know that these are there to help you detect the texture of objects, that they give us a fine point discrimination, and that we have a lot of these on different parts of the body, like the tongue, fingertips, but not so much on our back. Again, Ruffini end organs as well. These are primarily in the lower dermal layers, and this is sort of the deepest layer, the Pacinian. Ruffini is somewhere in between. These Ruffini end organs are not necessarily in the palm of your hand, but in the dermis or the deeper layers in your fingertips, and these respond to continuous touch or pressure. And that's the way that I'm going to be asking you to recall back, that you remember or have learned this material about being able to identify if we have this type of sensation, what type of sensory receptor system might be activated, if it's in the fingertips, for example. So I want you to be aware of this, that you will get a couple of questions based on the 4 or 5 slides that we've just covered related to how these different sensory receptors are found in different parts of your body and why all of this is important. Do you have any questions about this before I go on? Okay. So this is a really busy looking slide, and I need to know a little bit about what you have already learned in some of your biology classes. You guys have learned about muscle cells, right? Somewhere in your first year biology class. Do you know what motor neurons are? Do you know what muscle spindles are? Kind of? Okay. I'm going to make this not as complicated as it is here. We have 2 different types of muscle fiber. One which actually causes muscle contraction, and then we have this other gamma motor fiber with a motor nerve ending coming into it that is found inside the belly of or the big part of your bicep in your arms. Okay, it is not actually there to cause contraction. So this part here actually doesn't contract; it just sits there. And we're not going to go into it in a lot of detail, but one of the things that it does, and these 2 things go together, it tells you a little bit about what your arm is doing in space. So it provides information about the length of your muscles, and it's involved in what is known as the stretch reflex. Can any of you guess what the stretch reflex is? Obviously, it has to do with stretching. So it's like when you're doing the downward dog in your yoga class, you're activating your stretch receptors. Yes? Probably not, actually. Can you tell me what the stretch reflex actually is? You use it every single day if you're a commuter. Who has something relatively heavy? No one. No one has anything heavy. Okay. Let's see if I can briefly. No, maybe not. Oh. Where water bottles go to die. Can I get someone to volunteer for me? It's nothing terrible. Yes, you're flexing your muscles. That's a good sign. I will take you as a volunteer. Thank you. Okay, so this is going to be a demonstration of 2 things. One, muscle spindles, and this is the stretch reflex. Could you put your hand out like you're about to get 5 from me or something? So this is the stretch reflex. So this bottle didn't drop. What ended up happening is, sorry, and your name is Jimmy. He's feeling the weight on his hand, and his muscle is actually contracting to compensate for this. That is the stretch reflex. And what this does is it says that, you know, my muscles are getting longer. It's being stretched. Your muscles don't like being stretched. If you ever tried pulling someone's arm, their natural response is to pull back. And the stretch reflex that Jimmy just demonstrated, there's another aspect to this. So if I put this in his hand, he adjusts right away. So this is the muscle spindle. It knows the muscle is lengthening, and it's contracting to make sure that that is staying there. Thanks, Jimmy. Thanks. Okay, awesome. Sorry, I don't do the whole handshake thing. Thanks. Okay, so everything else, don't worry about it. I don't need you to memorize all of this. But but I do need you to know that muscle spindles found in your biceps, in your triceps, and everything else is there when your muscle has a load on it, and that load causes your muscles to stretch, and your muscles contract back in response to the stretch. This weight is stretching my muscle. My muscle is contracting back. When the train is moving around and you keep moving your legs, or you're contracting your legs in response to that, it's also the stretch reflex. Keep in mind one other thing we saw Jimmy do, or Jimmy not do, when that happened. And it goes along with this muscle spindle in the bicep. So if you saw what he did, one of the things that you should have been able to see is that he stopped it from falling. He adapted to the weight very, very quickly. That's one of the fastest reflexes you have in your body, as a matter of fact. And one of the things that also you were able to see him do is he adapted, and his muscle contracted back so that that extra weight that he wasn't expecting was maintained, but he also didn't over contract. Wouldn't it be a terrible thing that every time your muscle contracted, like when you get a weight on it, you actually like overcompensate and you smack yourself in the head? We didn't see him do that because at the same time that the muscle was contracting, we also had a number of proprioceptors found in the joint at the level of the tendon that actually said, there's increased tension on my arm. There's increased tension, your muscle is contracting, you should stop. There's going to be too much tension. And this Golgi tendon organ proprioceptor actually responds appropriately to make sure that you don't over stretch your over contract your muscle back, because that is responding to this increased tension on your tendons. Okay, so keep in mind again for your term test about the muscle spindle, the stretch reflex that we saw demonstrated, and that associated with it, these Golgi tendon organs, which are these proprioceptors, they tell us about joints, are also being activated at the same time in that stretch reflex. Okay, so here's where things are going to get a little bit more confusing. So what we've done so far is we've looked at the first side of things. We looked at all the things that cover your skin, and we talked about the sensory system on your skin, and that was the left side of the diagram that I started out with. Now we're going to move to the right side, the special senses, the things that I mentioned that we were going to go through, that your brain has to be able to decode things. In order for things to be decoded, there has to be a way for a chemical or a sensation or something else, or light, to be converted from its natural form into something the brain recognizes as electrical activity. And this is where we get into this idea that these sensory receptors produce what is known as a receptor potential. And this involves having both primary receptors, and this is where things will get a little bit confusing, so I'm going to try to keep it as simple as possible, as well as secondary receptors that actually cause the release of neurotransmitters. Now, the best way to actually explain this is in the next few diagrams. So if you have questions at the end of those of the diagrams related to primary and secondary receptors, then let me know. But you'll see that in every case, when a stimulus activates a cell that is responding to a chemical, light, etc., it causes depolarization, which is a generator or receptor potential, and then somehow it sends a signal to a secondary axons that will then convey the signal to the rest of the body. So here is an example at the top of a mechanoreceptor. So this mechanoreceptor is responding where we have pressure being applied on your skin. And if you take a look, this is that Pacinian corpuscle that we looked at earlier. So we get mechanical stimulation is pushing down on your skin. Eventually, it will stimulate this. It will activate a number of channels that are found here, and these sensory receptors that are found in this region will then create enough of a change in the sodium channels to cause this part of the dendrite to start to depolarize. It starts to create what is known as a sensory potential, or a generator potential. And then if it reaches threshold, it will then send an action potential back down this sensory axon. And this sensory axon will then cause neurotransmitter release onto the next neuron, just like we've looked at previously. So that's one type of scenario. And this is where we commonly find things going on in the skin and everywhere else in the body related to those sensory receptors on the skin. Another way to look at it, though, is this type down here. And this is what we're going to look at for the specialized senses, things like smell, things like taste, and other things. A chemical stimulus, whether it's in the air or in the food that you're eating, will bind to, in this case, a taste receptor. And when the ligand that is binding you to taste something, the binds to the taste cell, that will open up channels, just like we've seen before. This taste cell will depolarize. This taste cell, as the primary sensor, will then release transmitters, which will then activate this sensory neuron in the ways that we've seen it before. And this is sort of the overview that I want you to have, that we have these different mechanisms of activating and sending an electrical signal back into the nervous system in response to the appropriate stimulus. Okay, and again, don't worry about all of the details. If you want to go back and read through things on the left, it's just an overview of how we convert a chemical stimulus into electrical energy, which is what your brain recognizes. And again, how we can change mechanical force into action potentials at the top. And those are the basic categories that we're going to look at. Vision is a little bit different, and we'll get to vision separately from this. Any questions on that before we go on? Okay. So this is one of those slides that I'm going to spend a little bit of time on, because I think we need to know this, and we will go over these concepts again a little bit later on. I don't really call it accommodation. Your book calls it accommodation. I'm more used to calling it adaptation. And one of the things that happens, so when you first sit down, you're aware of your butt hitting the seat, and you have that sensation. But you're sitting there right now, and you probably aren't even aware that your butt is on the seat. And this is a form of adaptation. You've adapted. If you've ever from high school, or you guys a couple years ago. Yeah, so I had an opportunity, unfortunately, to visit my daughter's high school, and I'm walking through the halls of my daughter's high school, and these guys are standing by their lockers, as I guess they're still doing. Way back in the day, we used to do the same thing, but they bring out bottles of Axe body spray. I don't know why they do that, why they keep so much, and they're spraying it in the hallway. I happen to walk through that, and I get hit with all these different odors from Axe body spray. But after a while, and you smell that right away. But after a few seconds, you can't even tell that that's there anymore. That's a form of adaptation. And our receptors can be either rapidly adapting, or they can be slowly adapting. Okay, and again, different ways to look at it, but I want you to be thinking about adaptation, where the response is still there, the stimulus is still there, but your nerves are not responding in the same way. And these slowly adapting tonic receptors, so slow adapting are also called tonic receptors, are ones that really don't stop firing when the stimulus is still there. So for example, if you took that scenario I just gave you, if I was still able to detect Axe body spray after like half an hour, like, wow, it still smells really bad, then it would have been a case of slowly adapting tonic receptor activity. But in fact, what happened was I rapidly adapted. That stimulus is still there. I accommodated it really quickly. I couldn't even tell that that stimulus was still there. My neurons weren't firing even in the continued presence of that stimulus. So I'm hoping that you'll be able to keep these things separate and distinct. It doesn't only have to be chemical receptors. It could also be proprioceptors, which are the examples that are being shown here, that you're aware of, like your body sort of position, or after a while you're not even aware that something is in the position that it is. Okay, so you'll have to tell me in the next few slides that we go through, how many of you have had like a primary anatomy course? You've had gross anatomy somewhere. Any gross anatomy? A little bit? A couple of you? Okay, so this is going to be fun, or maybe not. I'm not so sure. Okay, so there's a couple of ways that we can approach this. I want you to learn the anatomy, but I want you to learn it in a way that is somewhat meaningful for you. I told you early on that the brain is not just a jumble of different nerves, just random. And we're going to go through the structure of the brain in a few minutes. It's the same thing with the spinal cord. When you have signals being felt in your fingertip, for your brain to be able to process it, that signal has to be transduced. There's pressure, a mechanoreceptors being activated, action potentials have to fire down an axon, but there has to be a highway, there has to be a roadmap from all the things from my hand going up into my brain to go along the same sort of route. An anatomical nerve tract from my hand should be kept together, in other words. Like there's no reason for signals from my foot and signals from my hand to be going in the same sort of track. And so one of the things that we have in the spinal cord, because it is the conduit that we use to convey information up into the brain, we have very, very specific routes or very, very specific roadmaps to where axons, and these represent axons, many, many nerve bundles going up and down the spine in the same area, to different, from one part of the body up into the brain. And this is what we're going to try to cover without a lot of anatomy background in the next few slides. Because you don't have a lot of anatomy background, I'm not going to go into a lot of detail. And there are some slides that I'm just going to introduce concepts to you. And there are some slides that I think all of you leaving a second year anatomy physiology class should know. So I'll concentrate on those. So let me orient you just a little bit, because each of these tracks we're going to go into in a few minutes in some more detail. When we talk about the spinal thalamic tract, for example, this is where when we feel pain, that pain information is being sent. All the axons together are being sent, conveying pain information from the periphery, your body, up into your brain in this spinal thalamic tract. And that's one example. This is the spinal cord that we looked at before. And I told you that one of the things that you need to know for your term test is some of the basics of the spinal cord. So if you were to take a look at this, and this is the spinal cord, if I'm pointing here, what is called? So this is the ventral root we talked about before. This is where we have an effector motor neuron, and this is found in the spinal gray, which is why it's shaded in gray. We do not normally find axon tracks going in the spinal gray. We find it in the white matter out here in the periphery. Okay, so most of the tracks that we'll see are located in the periphery going in the white matter. Do you guys remember what this is called up here at the top on the posterior side of the spinal cord? So this is dorsal, and what sorts of things is it responsible for? So it's sensory information, and is it afferent or efferent? So it's an afferent, it's coming in. Sensory information comes in through the dorsal region, and then efferent motor activity leaves through the ventral root. That's in the spinal gray. We're going to spend time looking at the white region that we haven't looked at before in the individual nerve tracts. Okay, so again, another thing to help organize things, and it's very, very simple if you think about it, and it becomes important a little bit later on, is if you take a look at the word, it goes spinothalamic. And what happens is the first half of the word tells you where the information is starting, and then it tells you where it's going. So it's going from the spinal cord to the thalamus, going from the spinal cord, spino-reticular, to the reticular formation in the brain. So this is relayed to you here. The first half of the word spino, where it starts. The second half tells you where it's going to. And these tracks are going from the spine to the thalamus, the spine to the reticular formation, etc. It's a little different for the naming up here, but can you tell me what you think this means, spinocerebellar? So it's going from spine to the cerebellum. Exactly. Okay, perfect. So again, because you haven't had a lot of anatomy, we'll take a look at some of the basics of the anatomy, and I won't ask you to memorize the diagram. It's actually sort of an overview of what is here in bullet points on the right. And each of these sort of cross sections that you see here are represented for you here on the side in this particular diagram. And in green, you are looking at the different nerves as it goes up these different tracks. So this is the spinothalamic tract. So what this tract conveys information from the spine up into the thalamus is it contains all the information in your body about pain information, temperature information, light touch, pressure, tickle, itching, all of this is conveyed along this tract. So if you were to sever this tract, in just this tract, everything else is left intact, and you were to cut it, you would not be able to feel on the opposite side of the body any sense of touch, pressure, temperature, etc. So it conveys all of that information. I'm going to ask you to know about this 3-neuron system. It's a really important system, but for the sake of brevity, we'll sort of bypass all the 3 different neurons that go up. But the most important thing is that there is a primary receptor. It sends information. It's a free nerve ending because it's about pain or temperature. So we know that it's got to be a free nerve ending. Could be a light touch or pressure. It could be another mechanoreceptor. It sends information into the spine. It sends this information anatomically. This is found in the anterolateral part of the spinal cord, which is why it's anterolateral. That part I do want you to know, that the spinothalamic tract is found in the anterolateral. So it's lateral meaning to the side. And this is what you see here. It's on the side of the spinal cord. Do you know what it would be called if you went the other way? So this is lateral. If you go inward toward the midline, what would you call that? I think I heard it. Medial. So this is lateral, going inward is medial. So this is on the antero side. This is posterior, this is anterior. It's on the anterior lateral side of the spinal cord. Okay, so that is something I want you to make sure that you know in terms of the anatomy. And this is where the spinothalamic tract is. This is the information that it conveys, and it goes all the way up in this tract up to the thalamus, which is what you see here. So those of you who have had any type of psychology, physiology courses, why the thalamus? So the thalamus is a structure that we're going to see over and over again. Every last one of the senses, so touch, things like vision, things related to hearing, every last one except for smell makes a connection into the level of the thalamus, and it's sort of a relay center before it reaches other parts of the brain that you see up here. So we won't go into a lot of detail right now. We'll come back to that thalamus many, many times, and you'll see thalamus being shown in all of the diagrams related to these different types of tracts. Okay, so I'm going to ask you to know this one as well. So this is called the dorsal column medial lemniscus system. And this is sort of an old name. This is a tract that we use to carry information about the 2-point discrimination. So I'm going to ask you to think a little bit about that. What receptor types encode 2-point discrimination? Is it Pacinian corpuscles? Do you remember what it was? Merkel discs. So this 2-point discrimination, proprioception, pressure, vibration, all of these things are going to be sent on the dorsal aspect. And this is the dorsal region up here. And it runs up as a column. And then at the level of the brainstem, it then switches into what is known as a medial lemniscus. Medial, again, is in the middle. So now it's been running up the backside of the spinal cord, then switches into the middle region of the brainstem, which is why it's called medial. And a lemniscus just means that it is a tract. So I just want you to make sure that you know that the dorsal column medial lemniscus system encodes for this type of information. Again, because you haven't had a lot of anatomy, I don't care if you know like the upper region, like the gracilis versus the cuneatus above the thorax or below the thorax. That's important for you. This is the system, the dorsal column in the spinal cord, medial lemniscus in the brainstem, that carries information about 2-point discrimination, proprioception and pressure vibration up into the thalamus, and then from the thalamus up into the brain, up into the cerebrum. Okay, so I just want you to keep that straight for the time being. Any questions about what you need to know for this particular diagram? I'm just going to mention one thing here because it's going to come up in a future slide. Do you know what this word here means? Decussation. Any ideas? Or can you guess based on the picture here? It's a little hard to guess, but you might be able to. Yes?
Is it from dorsal to somewhere else?
Dorsal to somewhere else. Kind of. You're almost right. Almost right. So let's take a look at this. So let's say this is the midline, and we split it right down the midline, right? What do you Yeah, left side, good. Not a trick question. Like on the term test, if you get that type of question, just go for the obvious response. It's not a trick. What side is this tract up here? It's more on the right, and so this decussation is a crossing over. And sometimes the tract goes from one side to the other, and we call it a decussation. Okay, and this or decussating is where it crosses over. Okay, I'm going to skip the trigeminal. I haven't really talked a lot about these different cranial nerves. You don't have to know this particular slide, and someone remind me about that on Facebook or something else just to ignore this slide. The spinocerebellar one, again, I would like you to make sure that you go through and know, but mainly for it carrying information about proprioception. And what is proprioception again, just so that I'm sure that you're sure? Where your joints are in space. So information about your joint, your body is in space to the cerebellum, and a lot of it is you're aware that you're standing upright, or if you were to close your eyes, you might move back and forth a little bit, but you would be able to stay upright because of the spinocerebellar system. Okay, and again, that's all you really need to know from this particular slide. I'm going to skip for the time being the spinal olivary because we haven't talked about the olivary nucleus and we haven't talked about the tectum. If you've seen someone with spinal cord damage, they have a lot of things going wrong with them. One of the things that goes wrong with them is their sense of balance. In people with stroke, the same thing. And some of these different tracts are really grossly affected. And so even your ability to track objects in space and your ability to do simple tasks like when you go to the ophthalmologist and they ask you to follow their finger, for some individuals that have spinal cord damage, even though there's nothing wrong up here, because of the way in which the spinal cord sends information up to the tectum, which controls your eye movements, these reflexes can get affected in them as well. So again, we're going to skip this for the time being. I am going to mention this in passing, and it's important, and I want you to make sure that you're aware of this. We have been talking about information that goes up, goes from your spinal cord up to your cerebellum. It goes up to your thalamus. It goes up to your brain. That's ascending information. It's sending information up to your brain. It says that you've just touched the wall. That wall is cold, you're putting pressure on your hand. That is sending information up to my brain, and my brain is processing. Your brain can then decide to send information back down. There's a descending pathway that modifies sensation. And some of this, and you might be wondering, like, what does that actually mean, modifying sensation? One of these, and again, if you take a look, this follows the same naming rule. What do you think this descending path means in terms of the naming rule that we just talked about? Corticospinal. Where does it start? Cortex, and where does it go? Spinal cord. So it's a descending track. It goes from the cortex up here down to the level of the spinal cord. It follows the same naming rule. And one of the things that it does is it can actually release endorphins. And so when we're talking about the runner's high, this is that descending pathway that can actually modify the sensation of pain, for example. And this modification of sensation might be that sensation of pain through the release of things like endorphins. Do you know what other pathway is, what other thing this descending pathway is really important for? Like, you need it, you use it all the time? Any ideas? Again, you won't have to worry about it for the term test. I just want you to know that the descending pathway, corticospinal tract, might release of endorphins, modify sensation, the bullet points that are here. But again, because it's physiology, because it should be relevant for you, where else do you modify pain? Not just like when you're running. What else do you do? You guys don't have a lot of pain in your lives? No? Okay, so should I give you more pain in your lives, like on your term tests, for example? No? That wouldn't be nice. So what sorts of things do you think might be related to the modification of pain responses? Yes?
We like injure another region. I mean, to induce greater pain, you avoid the pain in the region that is initially inducing pain.
Not sure I follow. Maybe. Yeah, at the back.
How multiple sensations to like relieve pain.
Multiple sensations to relieve pain. Okay, sure. So that's done at a local level. It's not done on a descending track level. So you're talking about if I have like a lot of pain receptors in my thumb, and I have a lot of I also have a lot of pressure mechanoreceptors in my thumb. I have both, right? It's not just like I have one or the other, and I smack my thumb. I feel pain because like I've activated the A alpha or the A delta pain pathways, the myelinated fibers. What do I do with my thumb as soon as I've hit it? Sorry?
Like cold water or ice.
Cold water or ice. Okay, I have no cold water or ice right now. Put pressure on it? Oh, that would hurt. Well, what do you do when you've like just hit your finger with your thumb? You shake it? Honestly, you guys don't like suck your thumb, like put it in your mouth or like put it under your arm or something? No? No, okay. You've just been your prof wrong. Good job. But you can stimulate the cutaneous receptors and cause presynaptic inhibition of the pain pathway, which is what you're talking about. Not what I'm talking about here. Have you heard of the placebo effect? You can actually cause someone to have less pain by releasing endorphins, by having them think up in the cortex that, you know, that tic-tac that I just gave you, that's going to make you feel better if you have a headache, and if you take it, you might. And that placebo effect uses the same descending pathway where you might release different neuromodulators like endorphins. Okay. So I'm going to ask you to know this particular slide because it's a really important overview of the different brain areas that I'm going to start to go through. And again, I mentioned earlier, this is a slide that you should probably make sure you go over on the weekend if you happen to see Drake showing up. And I think next week we're going to have guest appearances by Beyonce as well, just because we want to make sure that we have equal representation of the sexes within our course. If you were to take a look at the different areas of the brain, it's not just a random assortment of different groups of neurons. There is a structured order to how the different regions of the brain respond to different types of stimuli. We know that, by the way, from Wilder-Penfeld. When he starts stimulating this area of the brain, like the inferior surface of the frontal lobe, then people start smelling things even though there's no odor in the room. And we know that different regions are there to process different types of sensations. And we're going to go through each of these, and we're going to talk about the primary somatic sensory area, which is the postcentral gyrus. And I don't think we've had a lesson on sulci and gyri yet, right? Does that word mean anything to any of you? Sulci, gyri? The folds, right? So there are hills and valleys, and the hills, the things that are thrown up, like a hill, are the gyri, and the things that go down, the invaginations, are the sulci. And this is the post-central gyrus, and I'll get to what that means in a moment. And this is the area of the brain that's involved in making sense of all the different things that are coming up through the dorsal column medial lemniscus, through the spinal thalamic tract. It is there to make sense of all of the different sensations from your skin, to give you an idea like what's really going on. Okay, if you had a stroke up in this region, you wouldn't be able to make sense of all of the things that are coming up into this area. The part of the brain that is responsible for taste is the inferior end of the postcentral gyrus. And again, I'll go through the postcentral gyrus in a moment. The part of the brain that's involved in you being able to detect different smells is the olfactory cortex that we find on the inferior surface of the frontal lobe. I'll show you that again in a moment. Visual cortex, I'll just jump to at the very back of your brain, the occipital lobe, and the primary auditory cortex, which is on the temporal lobe, on the superior part of the temporal lobe. And again, the brain is kind of neat in the sense that this is the primary area. This is where things start to come in, but the areas immediately around it, they're neighbors. The neighboring regions, which we call association areas, they're like geographically right beside the visual cortex. We have the visual association area. It's the part of the brain that surrounds the visual cortex. This is where we can actually start to get more complex information being encoded. Okay, so again, just to give you an overview, this is that visual cortex at the very back of your brain, the occipital lobe that you see here. Immediately surrounding it are the visual association areas. So you might, oh yeah, I've seen that person before somewhere. I think they used to go to my high school. That would be the visual association area. Being able to see the person would be there at the visual cortex. So this is the primary auditory cortex, where you can have the temporal lobe. This is the temporal lobe. And then we have your ability to hear things, to process sound, and to hear different voices, to recognize different voices. We would make use of the auditory association areas right beside it. So neurons here get all the information from your ears. They detect changes in hearing, but then in order for you to make sense of what you're hearing, I think that's probably my mom's voice on the phone right now. That involves the nearby auditory association area. I'll just jump to this area up here at the top, and this is what is known as the central sulcus. It is the part of the brain that actually divides the frontal lobe. So everything in front of it, anterior to it, in the frontal region is the frontal lobe, and everything behind it is the I don't think it's indicated for you on this particular slide, so you don't have to worry about it. The thing is, it looks really cool on this diagram, right? But the reality is, if you were to look at a brain, it's not going to jump out at you, and you're not going to be able to say, oh, that's like the really big central sulcus. They all the same, to be honest. There really isn't something that you can use as you have to be a really skilled neuroanatomist to be able to find this central sulcus, which then helps you find, for example, the somatosensory cortex, which is behind it, and so we call it the post behind post. This is the central sulcus, and this is a gyrus, the sort of the hill part, and the pre-central gyrus up here is the motor cortex. So this controls your. It's the part of the brain that actually controls your arm, your legs, and everything else. And we're going to come back and spend some time on that as well. You guys are, again, you're gonna have to remind me, not so big fans of labeling figures, diagrams, out of the question. No? Thumbs down? Thumbs down. Okay. All right. Okay. So I'll ask you to make sure that you're comfortable with this particular slide then, rather than looking at it sort of in this particular format that you see here. Why no diagrams? I have great diagrams. Like, they're pretty. You'd enjoy it. It'd be like coloring things in. And, no. Okay. Okay. So again, you don't have to worry about all the details on this slide. This is sort of going back to this concept. Even with the brain, not just the spinal cord, moving things toward the midline is medial. Moving things away to the side is called lateral. And we do the same thing in the brain. This is what is known as a coronal section. So if I'm standing right in front of you and you were to slice through my brain this way, this is the coronal right-hand side, and then there would be another left-hand side that mirrors this. This is what is known as a homunculus, or a little man, and you can see that if you were to look at it from the medial part of the post-central gyrus all the way through to the lateral part of the post-central gyrus, you would have every part of the body mapped out in here. So for example, if I were Wilder Penfeld, I was a neurosurgeon in the 1950s, I had my patient in the hospital, they're wide awake because the brain doesn't feel anything, and you need to know if you're hitting a part of the brain that's known as the eloquent cortex. You don't want to leave them not able to talk or anything else. You can take an electrode, you stimulate this area, and this is what he did. He mapped out the homunculus, and so he would have this patient, he would be able to identify the post-central gyrus, and then he would use an electrode without damaging the brain, and he would stimulate this area, and he'd say, what do you feel? And they might say, you know, it feels like something is pressing up against my hand. Doing so, he was able to map out that this is what the different areas of the body on the brain map out to. So if he went a little bit further and he stimulated here, the patient would say, wow, it feels like something's pressing up against my hand now. He'd be able to very eloquently different regions that are involved in sensation in this post-central somatosensory cortex on the gyrus. I think if you were talking about, this is referred pain. I'm just going to skip to this, and then we'll take a break immediately after. This is where your body sometimes gets a little bit messed up. And this idea of referred pain is one where your body is used to the signals that it normally receives. Okay, so the best way to try to describe this is, if for example, I'm continually touching things with my right hand knows, for example, that I'm usually going to be touching the whiteboard, so it gets used to the same sensations over and over again. This idea that the same part of the body is normally stimulated is this idea of referred pain. So when we have a heart attack, we have visceral receptors that actually indicate that your heart, for example, is feeling pain. There's something going on with the heart. However, normally your heart's not in pain. It might be the very, very first time that you've actually experienced pain in your heart, and your heart is the one that's actually experiencing pain. The heartache, the pain. Okay. I feel that a physiologist should write a song about that somehow. I'm not sure how exactly, but what do most people feel when they have a heart attack? They don't feel like, that's weird, something inside my chest is like feeling terrible. What do you normally associate with a heart attack? Yeah. Yes, so you might have tingliness in your left arm, or you might experience, because your arm definitely feels things all the time, right? You might feel things like a tightening in your chest because you feel things with your chest all the time. And this is this idea of referred pain. Your skin is used to feeling pain. You get pain on your skin all the time. Your organs usually not so much. And so the only way that your body makes sense of this is to say, oh, you know what? Your arm hurts, but in fact, it's really your heart that is feeling the pain, because the same spinal segment, the same area that's sending information up to the brain, is now being stimulated by a different organ that it's never seen before. It's like someone in through the door and you've never seen them before. You just kind of assume that they're part of this class. They may not. Assumption. It's the same thing your brain is doing. They're just assuming, well, okay, I've never had information coming in from the heart before. Since the nerves are also coming in from the skin, we should really say that the skin, the part on your arm or in your chest, is the actual part that's hurting. Same thing for your, for example, if some of you have had a problem with your gallbladder, I don't think any of you here, because you're all really young, but if you get older, for example, and your gallbladder starts hurting, you're actually feel pain referred like around to your back. You might feel it in different areas of your. Okay, so this is this idea of referred pain, and the most important point here being that it's because both the organ and the region of the skin that you normally associate with pain are being innervated by the same spinal nerve segment. Yeah, do you have a question? And then we'll take a break.
Yeah, it's the same idea of going with phantom pain.
No, not exactly. Not exactly. And I think it's the next slide, and we'll cover that when we come back from break. So I think now is a pretty good time to take a break, and then we'll try to get through as many slides as we can. You haven't had any
Question in regards to our assignment.
I'm just reading online that instructions and stuff, and that's to do with websites.
Doesn't have to be NHS, so it can be like American Journal of Physiology.
Yeah.
That's pretty decent.
Yeah. So, because I wanted to ask you, I'm thinking, like, actually,
Can the article you use in the notes, because that's what they're turned in.
yeah.
So if you come up as well, you've done this.
Yeah, no, I understand that. So, even different courses, chemistry,
Turn it back here where you submitted.
yeah.
Just say, you submitted this here, it looks like it's in there. Like, you say general stuff, or not?
It has to be different. Right.
Different submission. I don't care if the topic is the same. I don't even care if the paper is the same. It can't be what you used before.
Yeah, obviously, I wouldn't copy and paste it, but I can work on it, because that's what I can copy. But definitely, there are some things
So, I mean, just start with description. Okay, folks, if you don't want to get going. So this particular slide, I'm actually going to point out two different things related to phantom pain and chronic pain that I'd like you to be familiar with for your upcoming term test. Your colleague had actually asked about phantom limb pain earlier on. And for those individuals that have their limb amputated for a variety of reasons, whether they're war veterans, if it's a disease condition like diabetes, etc., I haven't really encountered, to be quite honest, a lot of individuals who've had phantom tooth pain, but I think your book points this out as well. Those individuals, even though there's no possible way for their arm to hurt because their arm is gone or their leg is gone, they still feel like their arm or leg hurts, and it hurts a lot. They're not really sure why that occurs. One possibility, and this is the theory that was put forward by V.S. Ramachandran down at the University of California at San Diego, that your book highlights for you, is what is known as the gate control theory of pain. And we talked very briefly about this, that in theory, if your digit or your hand or your limb is in pain, if you massage it or if you were to stimulate the mechanoreceptors, it can actually turn off the pain receptors. So you hit your thumb, you put it in your mouth, the mechanoreceptors stimulate, they turn off the pain response, then you feel a little bit better. Not a lot. It's not like your thumb is going to magically heal itself. But in theory, when that limb is gone, that inhibition, the ability to turn off a pain signal, is also gone. So yes, the limb itself is gone, the pain receptors in the limb are gone, but so is the possibility that maybe this gate control, where you have mechanoreceptors in your arm being able to turn off a pain signal, are also gone. This inhibitory effect, in other words, after amputation might be lost. And I want you to be able to equate this gate control theory of pain, and it's called gate control because all pain signals are gated at the level of the spinal cord. Anyone who has spinal cord damage, they don't feel anything. And it's because the spinal cord really ultimately controls what gets up into the brain. And if you can turn off a signal at the level of the spinal cord, it's gated or turned off at the spinal cord. And phantom limb pain is thought to involve a loss of this gate control of pain responses. Unfortunately, in some individuals, and again, it's becoming more common for whatever reasons, you don't actually have to have a direct tissue injury. Your leg, your arm, might have healed itself completely, and many years later, after your arm in theory has completely healed after being broken or after having a burn, you might still feel chronic pain. And this form of chronic pain, it's believed to involve a mechanism that we'll go through really briefly in a few moments, but it might be that some of the synapses and some of the brain pathways, either in the thalamus, the sort of the gateway, if you will, up into the brain, everything stops there before it goes up into the cerebral cortex, or something in the cerebral cortex just really goes wrong. And we're not really sure how to regulate this, to be quite honest, and chronic pain is becoming a real epidemic in most of the modern world. And so we don't really know how to control it. This whole brain regulation at the level of the cerebral cortex is not something that is something that we can control. Typically, we think things like migraine, back pain, but it can be like a whole host of things that cause chronic pain. And you can tell that it also brings on other things. So if you have a migraine, back pain, chronic pain that lasts for a very long period of time, for example, some individuals will have chronic pain related to childbirth and delivery. So they'll give birth. Labor pain is very, very common. They did not have an epidural at the time. And many, many years after childbirth and delivery, they will still have pain, not just back pain, but all sorts of pain. And they will also have some of these other symptoms like depression, feelings of helplessness, hopelessness, etc. And chronic pain is really something that we're not good at understanding right now. So I'm going to give you a little bit of an overview, and we'll come back to skeletal muscle after the term test. And the skeletal muscle system that we're going to look at is mainly the voluntary movements involving either upper motor neurons or lower motor neurons. And I'm going to come back over that later, so I'm not going to emphasize this for your term test because we're coming back after your term test to the skeletal muscles later. For the time being, I want you to know that there really are multiple levels of movement. So you can move just based on your spinal cord. Like some of you will have learned about the patellar tendon tap. So you sit, the doctor hits your kneecap, and your leg moves, right? It didn't require you to think about you moving your leg. It's at the level of the spinal cord. It's a reflex. Reflexes cause you to move. Your ability to walk actually is a series of reflexes that you've developed over time. You're not born walking. You have to learn how to walk. And these reflexes start off at the spinal cord, but then move up into the motor system in the brain. And so the motor system is something that we'll talk more about later. This is a slide, though, for the time being, that I'd like you to make sure that you are with. And again, it's not just this slide, but the steps in voluntary movements. You have to understand that your brain has to initiate this. When you are thinking about doing something and initiating a movement, you have to plan it up in your brain. And yes, there's a motor cortex. Do you guys remember which gyrus it was located on? Is it pre or post? So it's the pre-central gyrus. It's closer to the front of your brain where you control motor movement. But the reality is, even before you stimulate that part of your brain, you actually have to plan it in the area of the brain called the premotor cortex. Okay, so the planning stage actually begins in the premotor area. And you might not think about it, but the reality is if you're heading toward the door and you are planning to open this door, the premotor cortex, even before you have moved your arm, which is the motor cortex, and turned the knob, which is the motor cortex, your premotor cortex is already planned out that you have to grasp, turn, and do that. You don't think it's done all of that, but it has. And these are one of the steps: premotor planning, then activation of the motor cortex, then activation of these upper motor neurons that form this descending track. It goes from the brain down, so it's a descending track. And then they will activate the motor neurons that directly innervate the skeletal muscles. Skeletal muscles are always voluntary. There are other things, again, that your cerebral cortex can interact with, like the basal nuclei, as well as the cerebellum, in terms of the coordination as well as execution. But the reality, again, is you don't have to have a cerebellum. There are lots of people in the scientific literature that have had what is known as cerebellar agenesis. So they started off being born with a cerebellum. Their cerebellum degenerated. The rest of the brain was left intact. No one even knew that their cerebellum had disappeared. They're perfectly fine. They're just a little bit clumsier because they can't coordinate well as an individual who has a cerebellum. So these are the different steps. These are the accessory structures in the brain that I'd like you to be familiar with in terms of voluntary movement. So again, this is all related to, this is the central sulcus. In front of it is the pre-central gyrus. Behind it is the post-central gyrus that we talked about in green. This pre-central gyrus related to movement. And in many ways, it looks exactly like the mirror image of what we find on the other side of the sulcus. Do you remember there was a little homunculus that we saw here? There's another homunculus that Wilder-Penfeld mapped out in the same way on the pre-central gyrus as well. In these premotor areas that you see here in lighter purple to coordinate with the darker purple that you see here, these are where you start to organize the different activities. The motor cortex is there to move the muscles, to actually do everything, but the premotor areas have to plan like what you're going to be doing. You might not even be aware that you're doing that, but if you lesion this area, you have a stroke in this area, your arms may be able to move perfectly fine, like you have no muscle weakness or anything else, but you might have difficulty being able to open a door if it has affected your premotor area. And again, even before that, in another association area, you might be thinking of like, okay, I need to plan to do something in order to exercise. I need to plan way ahead of time, and that involves other accessory areas in the prefrontal cortex as well. Again, this topography, you can see it looks very, very similar, not identical, but very similar to what we saw earlier with the homunculus in the somatosensory area. This is just on the opposite side of the central sulcus. No need to go into a lot of details. Let me ask you one question though, because I didn't do it on the somatosensory cortex. What do you think of the homunculus? It's not continuous, and it's not in the order that you would normally see the body mapped out, right? Do you notice anything else about it? Nothing? Yes, at the back. There's no ear? Okay, I guess there isn't an ear. Okay, yes, I'll buy that. Not what I was looking for, but okay. It's like writing in the answer F. No choice F, but go ahead and write it in. Yes.
It's not proportionate, like some areas are bigger than
Right, so it's not proportional. Some areas are bigger than others, but think a little bit about that. What do you think that actually means? So you're 100% right. Great observation. Definitely compared to like your back, which is up here, the motor cortex that controls like your lips and your tongue is much, much bigger. What does that mean? More neurons that are devoted to actually carrying out the movement of those particular areas of the body. These are all related to muscle groups, and these are all related to movement of these different areas. And you have a lot of neurons in this area of the brain to control things like your lips, your lungs, because we talk a lot, for example. We have a lot of neurons that are there to control the movement of your hands because we use them a lot, but we don't need as many neurons to do things with our back, which is shown up here, or with our knee or our hip. Okay, so great observations. The nervous system is sort of logically put together, and it's something that, again, I'd like you to make sure that you're aware of. Since we had indicated earlier that we're not really big on anatomy because we haven't had a lot of it, I'm going to just mention a couple of things. I'm not going to ask you to know as many details as I had originally sort of mapped out for you. We have two different types of motor tracks. One is a direct pathway that involves the pyramidal system, and these are the direct pathway pyramidal systems that you see outlined for you. The corticospinal tract, again, the name makes sense, goes from cortex down to the spinal region, and then the red nucleus or the rubrospinal tract, red nucleus to the spinal cord, reticulum, the reticular formation to the spinal cord, and the vestibular apparatus into the spinal cord, and the tectum into the spinal cord. There are direct and indirect pathways. The only thing I'm going to ask you to know is what's on these bullet points. These direct pathways are there to control things like muscle tone, how fast you do an action, and the precision of skilled actions. For you to be able to play the piano, you need to be activating these direct pathways, these direct motor pathways to move your fingers, to have that speed and control for precision movements. These indirect pathways are not as important because you need less, or not, sorry, not as important is not correct, but less precise movements are dominated by these indirect extrapyramidal pathways. Okay, and again, we're going to come back to this, and we're going to come back to this when we talk about muscle and the control of muscle. Can you think of a disease condition for the time being, and then we'll sort of skip through this slide. Can you think of a disease condition that you are aware of that actually affects movement? What diseases do you know that affect movement? Yeah?
Paralysis.
Sorry?
Paralysis.
Paralysis, okay, so paralysis is because you've completely cut off these pathways. There's no way for it to go down. And control muscles, absolutely, yes. Parkinson's disease is one where these different pathways are affected at the level of the substantia nigra, and it's part of this control pathway that you see here running through a number of direct and indirect pathways. And we're going to come back to examine Parkinson's disease, its role in controlling skeletal muscle, why you have different types of lack of movement in Parkinson's. Can you think of the opposite condition? If Parkinson's is I can't move, what is the opposite condition called? Do you guys learn that at all, or have you heard of it perhaps, or volunteered in an organization that deals with it? Any ideas? Parkinson's, you don't move. Absolutely correct. It involves the substantia nigra. But can you tell me about another condition where you move too much? Yes.
Actually, I was thinking multiple sclerosis.
Okay, so multiple sclerosis we did talk about. Demyelination, axons can't actually convey a descending signal. And that's lack of movement or lack of coordination. How about moving too much? Yes.
I remember for some of the medicine that schizophrenic patients used to take before.
Yep.
The earlier medications, it would cause them to have jerkish movements.
Yep. It definitely did.
They are out of control. They didn't want to enact those.
Yep, definitely things like chlorpromazine, haloperidol, other things that activate the dopaminergic system. Dopamine, because it works at a number of these different synaptic connections, can cause overactivation. Sure, but it's not a disease condition. Well, it kind of is. Schizophrenia, you're right. It is a disorder. Yes. Tourette's, yeah, maybe. It's not really a motor condition. Have you guys heard? Yeah. ADHD, okay, another interesting condition. Not sure it's purely motor. Have you guys heard of Huntington's chorea? Have you heard of like choreiform movements where you have like your arms swing like really wildly? And those individuals, they have overactivation of the motor pathways. And we'll come back and talk about that a little later, and we'll come back and talk about these direct pathways for the term test right now. I don't want you to be spending too much time on this, because we're going to come back and talk about these when we talk about muscle systems as well as movement, and we'll come back and talk about corticospinal versus cortical bulbar tracts a little bit later on. And yeah, I don't think at this point we're not going to spend too much time on this. We'll come back and cover it after the turn test related to skeletal muscle. So apologies on this. If you want to read ahead or you want to go back and review it, when I come back to it, these are the things that I'm going to concentrate on. So you can just reference that for the time being. For the term test, which is what I'm trying to gear you up for, we don't really need to worry too much about this. Okay, including these indirect pathways, and I think we're going to skip through the basal nuclei, and I have to apologize. So again, this goes back to the cerebellum. You can live perfectly fine without a cerebellum. Many instances where people have been reported, they didn't even know they didn't have a cerebellum. They just went in, they had an MRI done. By the way, did you happen to know you don't have a cerebellum? It happens, and it's not as uncommon as people would like to think. And again, we won't worry too much about some of the other aspects about posture as well as coordinating movements. So I am going to ask you to know this slide because it is sort of an important slide. We haven't really talked about the overall structure of the brain. So we've sort of gone in a non-traditional way. Your book covers it in sort of a non-traditional way. We started off with a little bit of spinal cord, then we just jumped to brain, and we talked about different regions of the brain. And we sort of bypassed the thing that connects the brain, the spinal cord to the brain. And this is the brain stem. The brain stem is like really, really important. The spinal cord works its way up into the brain stem, and all of the different pathways that we talked about, the descending corticospinal, descending cortical bulbar, the ones that control muscle movement, work their way through the brainstem. So do the ascending, the sensory signals that go from the periphery up into the brain also go through the brainstem as well. And it makes sense because it's just a continuation of the spinal cord. The other thing to make note of, how many cranial nerves did we say we had in the last lecture? 13 or 12? 12, 100% sure, final answer? Yeah, okay. So we have 12. Yes, okay, good. So we have in the brainstem. So as we're going up the spinal cord, it then works its way into the brainstem, then it goes into the midbrain and pons, and then you get the cerebellum on one side and the pontine formation on the other. The vast majority of the cranial nerves, all of them except cranial nerve number 1, are located at the level of the brainstem. So it tells you a lot about what these different cranial, the importance of the brainstem. The brainstem is also really important because it controls a number of different reflexes. So reflexes that are vital to how you regulate your heartbeat, your heart rate is located here in the brainstem. Your blood pressure is controlled at the level of the brainstem. Your basal level of respiration. So when you're sitting here right now, you are breathing at a rate that is set by your brainstem at about 12 to 15 breaths per minute. Your brainstem has decided that that's the rate that you should be breathing. How you sleep is also regulated at the level of the brainstem, and this gets into the reticular activating system, and then we're going to get into this sleep-wake cycle. Whether or not you decide to vomit is also regulated at the coughing, sneezing responses, all of this. It controls a lot of your basal functions. In other words, you can live without a cerebellum, you cannot live without a brainstem. You would die instantly if your brainstem was damaged. And it's one of those things that you should really make sure that you are aware that your brainstem controls so many of these different pathways. Because your brainstem is also important in sleep, because it contains what is known as the reticular activating system, this is a group of neurons. Reticular just means network. It's a network of neurons that activates and turns on your brain. They're found at the level of your brainstem. And these are the ones that control how you wake up and when you go to sleep. And some of the reasons why we go to sleep are determined by all the senses that we get in. Since most of our cranial nerves that bring in information from the head, things related to sound come in through cranial nerves, your eyes, all of this comes in through your cranial nerves. A lot of that information gets processed from the cranial nerves at the level of the reticular activating system. So most of you, unless you've been staying awake a long time, probably won't go to sleep during the day where your optic nerve is bringing in information to this reticular activating system saying that you know, there's a lot of light out, so you're likely not going to be sleeping. And again, that information is encoded at the level of the brainstem. Again, one of the things that your book kind of gets right is that one of the last things the last sense that you lose before you actually go to sleep is your sense of sound. Your first sense that goes before you go to sleep is actually sleep. So most people can sleep through smoke in their homes if their home is going up. But the sound is actually the last sense to go as you go into sleep, and it's the first sense that actually gets turned on by stimuli. So again, most of the alarm clocks work on sound. It's not like related to light or anything. It's not even sense of sensation to cause you to sleep, all related to this reticular activating system. A lot of different sensory inputs, auditory input, sound, visual input, the amount of light going into your eye, senses of smell can wake up some people. It's a great way to wake up, for example, to the smell of coffee or to breakfast being prepared. All of these sensory inputs, along with emotional input from the limbic system that we haven't covered yet, all work at this level. This is the brainstem. This area in blue is the loose network of neurons, which is called the reticular activating system or the reticular formation that control how you go to sleep and how you wake up. Okay. All right. So we'll go through a couple of these brain functions. Then I'm going to take you through, I think, in the next few slides, different aspects of the sleep-wake cycle, a little bit about learning and memory, and then I think we're going to try to wrap things up somewhere around 6 or 77, which means I have a lot to cover next week, but I'm okay with that as long as we are staying on track to be able to get all the things in that's on your term test. Again, I'm not going to ask you a question about this, but I wanted to cover it just to make sure that you understood that the next few slides, if you're looking at it thematically, suggest we talk about the brain as this structure, and I've said that this structure is well organized. Most of you will know that the brain actually has both a left side, right side, and they're not equal. And one of those things in terms of higher brain functions that we know is not equal is speech. And we know that our brains are asymmetric because in the vast majority of individuals, including left-handed individuals, if you're wondering, your speech area is normally found in the left side of the cerebral cortex. And on the left side of your cerebral cortex, we have specialized areas, things like Wernicke's area. So this is where this group of neurons in this part of the brain on the left side of your brain actually can allow you to understand what someone is saying. So after you processed what someone is saying, if you want to respond back, you also on the left side of your brain have to activate Broca's area, which is also again lateralized or found on one side of the brain related to speech. So this controls the muscles that allow you to make those sounds that allow you to speak, also found on the left side. Again, for those of you who might be thinking about a career down the road related to speech-language pathology, one of the things that happens is some individuals have aphasia, where they have either or defective speech or language comprehension. That could occur for a number of reasons. Oftentimes, though, it's caused by a lesion. If someone, for example, has had a stroke, they were perfectly fine before. If you're a speech-language pathologist and you need to have them relearn how to speak and understand what is being spoken to them, this form of aphasia is something that they encounter quite often. We know from other types of studies, and again, you don't have to worry about the details on how to do a PET scan or a positron emission tomography scan, which is what you see down here. You inject a radioactive tracer that binds, in this case, I think it must be 14C glucose. It shows you which areas of the brain are more active. The higher red, yellow colors mean that this area of the brain is more active. And what you can see is if you see a word, if you're reading something, actually you're thinking about how to actually say that word. So you see the word, the visual cortex, and what part of the brain is this? Frontal lobe. It's the occipital lobe. The occipital lobe lights up first. Then we activate Wernicke's area for language comprehension. So you've seen it. Your visual cortex is activated. You're trying to understand what the word actually means. And then you activate Broca's area. There's a stepwise progression, in other words, in a temporal fashion where you get different areas of your brain lighting up, and it's all on the left side of your brain. Your left side of your brain is specialized, in other words. And then in order for you to actually say the word, the primary motor area is going to be activated as well. So it's not on here, but can you tell me what would the structure be in the brain that contains the primary motor cortex? Remember, is it precentral or postcentral? It's one of those 50-50s. How many of you say central gyrus? How many of you say postcentral gyrus? Don't put up your hands. Okay, it's precentral gyrus, right? It's the motor cortex. So again, we can see that there's a stepwise progression. There is a right and left side difference in the brain. And again, it's primarily related to sensory information, might be related to language as well. To be honest, this is still very, very controversial. I know it's in most of the sort of posters that university students love having in their dorms about left side artistic brain and right side sort of the mathematical brain. There's little evidence to actually suggest that it's strictly all or none, that your right and left side can both process math. In terms of lateralization, we can only see speech as part of that. And again, it's very controversial as to how this occurs. The only other thing I'm going to add, because we're going to come back to this a little bit later on, and it's a term in anatomy that we are going to come back to quite a lot. Do you know what the corpus callosum is? So I just told you the left and right side of the brain are a little different. So the language, Wernicke's, Broca's area on the left side. Do you know what the corpus callosum is? It's the left and right side. Yeah. Kind of. I think you are on the right track. So if this is the left side of the brain, and this is the right side of the brain, it's not like physically from one another. To be able to talk to neurons over on the left side. And a number of these axons that are sent from left to right, and again from right to left, they make up this tract known as the corpus callosum, tract that connects the two different parts of your brain, the left and the right side. And this tract that connects the left and right side is actually a really important one. Can you think of a disease condition that you might want to actually not have the corpus callosum around? You can survive, by the way, without the corpus callosum being intact. Any idea? Because again, I want it to be somewhat relevant for you, and you might have heard of it in other courses, or you might have heard of it in psychology, maybe in cell biology. Have you guys heard of epilepsy? So epilepsy is where the entire brain, instead of like functioning randomly, and this gets actually into brain waves and sleep really well, is all synchronized. Everything at the same time. For whatever reasons, the entire brain lights up at the same time. Usually, it starts in the temporal lobe for most individuals, and it usually starts on the left side, and then it synchronizes across the entire brain. And that becomes really, really dangerous for some individuals. And so what happens is some neurosurgeons actually cut these corpus callosum fibers. So if these neurons start to become overly active, their axons don't actually travel and they don't activate the ones on the left. And so this corpus callosum that connects the left and right hemisphere of the brain can be cut. And this is actually known as a commissure, or a axonal tract is another way to describe a commissure. And this corpus callosum actually can be deleterious in some instances, like epilepsy, where it actually makes both the left and right side of the brain active at the same time, which is actually not a good thing in something like epilepsy. So this is a series of slides that I'm going to ask you to know the details of the bullet points on the left. We can, on the surface of your skull, measure very, very small electrical activity based on millions to billions of neurons being active at the same time. So this EEG or electroencephalogram allows you on the surface of your skull to measure the brain's electrical activity. And it measures real electricity. We don't really feel very much on the surface of your skull, but these electrodes amplify and magnify the signals quite a lot, and we can place them on specific regions of your scalp in order to make sure that we know which areas are active. And when we do so, for most individuals, we can identify these different brainwave patterns that I'm going to show, including things like these alpha waves. And these alpha waves, in this case at the very top, mixed in with beta waves, these alpha waves can occur normally during rest. So if you're just meditating and you've got your eyes closed and you're just relaxing, calming down, you can induce more and more alpha wave activity. If you are concentrating on something, so imagine you are thinking about your term test. In theory, if you're really concentrating on your term test and your brain's really going at it, and I had an EEG, we would see more and more of your brain undergoing this beta rhythmicity, and it's a little clearer on the next slide. Theta is normally found in children, but we have it in adults too. So if, for example, I gave you one of those really weird multiple choice test questions where it was like, A is right, and then choice C is both A and B are right, and then choice D is none of the choices above are correct, and you're trying to figure out like what is the right answer that I should be putting down. And you're frustrated, then your brain undergoes this theta rhythm, and each of these theta rhythms, etc., and I'll just show you on the next slide, are bigger in amplitude. They get bigger. So alpha is relatively small, beta is a little bigger, theta is even bigger still, and the delta waves are the biggest still. Delta waves, though, we only see in very, very young infants, in severe brain disorders, in individuals that have lesions on parts of their brain, we can see delta wave activity, but most oftentimes in people that have no issues with their brain, at your age, for example, it's when you are in deep sleep. And these are sort of the changes that we see as you go into deeper and deeper levels of sleep. So at the very beginning, at a very light form of sleep called REM sleep or rapid eye movement sleep, you can see that the overall electrical activity is not very different than you would be at rest. In alpha or beta. And then as you go into deeper and deeper sleep, which is called NREM sleep, then you can see these waves getting bigger and bigger and finally forming these delta waves. When you go to sleep, you actually undergo, for a typical individual, let's say you are, I don't know, what time do you guys normally go to sleep? I know you don't sleep at 12 o'clock, right? Usually 1 or 2 o'clock in the morning, you're on Facebook, checking out Netflix, doing your whole thing. But let's say you go to sleep at around 12 o'clock. You will undergo sort of these different stages, get down into delta, and then you'll come back up. You'll go into a period of REM sleep for a little while, and it's very, very brief. Don't stay there very long. And then you go back down into the delta waves. And then as you get closer and closer to the time that you have to wake up, in fact, you go back into longer and longer forms of REM and spend less time actually in NREM or slow wave sleep. And when you're thinking about dreams that you have when you're sleeping, and if you're trying to remember them, the ones that you have during REM sleep out here are the ones that you typically think that you remember the best. So we have multiple REM periods, and we are interspersed at the beginning with NREM forms of sleep as well. Okay, any questions about that before I go on to about the next 4 slides, and then we'll call it an evening? Because the next 4 slides are actually really important slides that will be, for sure, on your midterm, your term test. How many of you have seen this in a psychology class about memory? You guys have all seen this, right? Can you explain it to me? What does this actually mean? So we're talking about this. You are trying your best to try to memorize this slide. How, according to this diagram, how should you try to memorize this slide? What are you supposed to do? Yes.
You're supposed to like repeat it to yourself multiple times. So like
Okay.
last.
So right now, in theory, if I could do it, and I don't think I can, but if I were to do it, can you tell me like what was on that slide? No. But you were seeing it, right? You had that sensory memory. That sensory memory is really, really short. It lasts about 1 second. What color was the one of the boxes? Purple? Pink? So purple, pink, okay, great. So this sensory memory is very, very short term. You don't hold it very long. In order for us to actually remember things, we actually have to convert this working sensory memory where you are seeing this. This is all your senses coming in together. You're hearing things, you're seeing things. It's all sensory memory. We have to work it into short-term working memory, the sensory memory. We have to repeat it, as your colleague said. That's the way that we think we learn the best, through repetition. Is that true? Do you think so? Yeah? Really? So I always have this conversation with my own colleagues, right? We're all neurobiologists, or I have colleagues who are in psychology. So you've just been sitting here for the last 2.5, almost 3 hours, right? How long is a movie that you guys watch over at Young Dundas? 2 hours, 3 hours? 2 hours, roughly? So what's the difference between what you are watching at Young Dundas? Because I have a screen here, all right? You guys are all sitting down. You guys were eating. You guys pay a lot of money to be sitting here, to be honest, almost as much as you pay at Young Dundas. But my guess is you remember more about the last movie that you saw, even though the conditions are pretty similar to what you're seeing here, on one shot, without you watching that movie multiple times, than you are here. What's different about that? You didn't have to repeat it. I bet you if some of you went to go watch, I don't know, hopefully none of you went to go watch Batman versus Superman, but if you did go watch Batman versus Superman, I remember from that. By the way, you always team Batman. But anyways, yes. Sorry? Okay, but aren't you paying attention here? Yes.
I find when there's a smooth connection of ideas,
Okay.
I tend to remember better.
You tend to remember better. Smoother connection of ideas. Okay. Yes, at the back. No, nothing? Did you have something?
Visual and auditory aids.
Visual and auditory aids? Like during your movie, you have visual and auditory aids? Like you have little things popping up and someone's up there with the laser pointer. Like this is over here is Batman. This guy over here, Kal-El. Like you don't have those. But you still remember like probably different scenes from the movie, etc., right? Yes.
There's a narrative.
There's a narrative. Okay.
So, like, if you were telling this like a story, it
Yep.
would, like, I think we'd probably remember it better because there's between one thing versus the other.
I think you're absolutely right. There's a lot of narrative, context, and everything else that you get this linear progression of a story. There's music, things that kind of capture your emotional attention. And the reality is, all of that helps you take working memory through long-term potentiation into short-term memory, where you remember things better than repetition. Because to be honest, even if you remember this short-term through repetition, like you go over your notes multiple times, write them out multiple times, re-listen to this lecture multiple times, to be honest, it won't stay in your memory as long as that last really exciting movie that you were really looking forward to, that you wanted to go see. Maybe on the weekend it might be Captain America 3 or whatever it is. So how do we go from here to there? There is a cellular mechanism that we think is responsible. If you can get that to click, if it works, then in theory, at least in theory, this long-term memory for different types of things like declarative memory, as long as there's an emotional content to it, actually you're not going to lose a lot. You don't have to lose a lot. You'll remember that movie for years and years at a time. Procedural memory is where you're actually doing something. Like it's the development of a skill. If you've learned to play the piano, it's likely that for the rest of your life you'll remember how to play the piano or ride a bike or do or go swimming, even if it's been years and years since you've last gone swimming. So why are some forms of memory different than others? We still are doing things to the brain. How do we get this consolidation, for example? How do we make sure that we're not losing things to make sure that we get more being pushed into long-term memory? And this is sort of the thing that I'm going to try to end with. We don't know. And these are the types of things that, for example, students want to know. Like, don't you really want to be able to walk out of here, in theory, and remember everything from this lecture so you don't have to go back and review it later? Because imagine how great life would be. I got it. I remember everything. My weekend is free. I don't have to do a single thing. But the reality is that's not the way that our brains work. But if we could, how could we push it so that we get more and more going into this particular pathway? And in fact, we know that our brains actually have the capacity to strengthen connections. And by strengthening connections that we call long-term potentiation, whether it's because we really like something, we really have an emotional connection with a movie, a musical piece, or whatever it is, a narrative, a context, whatever it is, we can get it from short-term sensory into short-term memory and then consolidation really, really easily. And we think that every part of our brain is capable of undergoing this long-term potentiation or LTP. This, again, involves this particular mechanism where we have glutamate being released in different neurons. And it's not shown here, but do you guys know what area of the brain has been shown to be involved in memory? What area of the brain is the most important for you in terms of memory? It is the hippocampus. You have two of these. We have right on the temporal lobes, we have this hippocampus. So it's a little bit bigger than your thumb. And this small structure is responsible for your learning as well as your memory. And we know that in the hippocampus, when you're trying to remember what's on this slide, or you're trying to remember what I just said to you in the last 5 minutes, when the neurons in the hippocampus release glutamate, if you are going to remember things, then this has to happen in the postsynaptic neuron that's receiving it within the hippocampus. Glutamate has to work its way across. Glutamate has to bind to its glutamate receptors. And then the channels have to open. Calcium has to enter through. And this is different. It's not the sodium that we talked about before. In this particular instance, calcium enters through, and the reason calcium is important in this case is because it will bind to molecules, calmodulin, and then activate calmodulin-dependent kinase 2, or CAM kinase 2, and CAM kinase 2 then makes these receptors much more effective. And this is why we think we learn. On that one shot, instead of just having things moving through, if we activate CAM kinase, it will make this receptor system much, much more effective. It's like you literally being able to walk out of here just remembering exactly what you've seen, because these receptors become much, much more efficient by being phosphorylated, and we also drive more of these receptors to the surface. That means that the hippocampus becomes supercharged, in other words, through this calmodulin CAM kinase dependent pathway, because next time that memory comes through in the form of glutamate, it's ready and waiting to be activated again. So I think I'm going to stop here, and we'll come back and revisit the rest of the lecture that I haven't covered. If I can make 2 suggestions for you over the weekend. I want this to be as smooth a process as possible. For those of you who came in a little bit late, the guidelines and the rubric for your written assignment are available under the contents folder. You do not have to follow the guidelines strictly. You don't have to pick only a Nature or a science paper. Any primary journal article over the last few years is fine. I'm going to ask you to pick a paper, if you can, and bring it in to chat with me or email it to me, and I'll be able to tell you, yes, that's a great paper, go for it, it's fine. The second thing is, I want you to study over this weekend just a little bit. So the least thing that you should do is to do the practice questions, see if you can answer them without looking at your notes, then try to answer them after you've looked at your notes. Those will be the same types of questions that you'll get on your term test. If you have problems after you've looked at your notes, then on Monday, you should come and talk to me. We will figure out a strategy, whether it's related to this or other things, on how to make sure that you do well on your term test. Other than that, have a great weekend. I'll see you all on Monday.